Hello everyone, it is a great honor to share with you our team's academic paper published in the journal iMeta. The title is, Macrophage-derived reactive oxygen species promote Salmonella aggresome formation contributing to bacterial antibiotic persistence. Professor Fang from the Biomedical Pioneering Innovation Center Biopic at Peking University is the corresponding author. The co-first authors are Dr. Xiao Chen, a former PhD student from Professor Bai's lab, and Kefan Fang, a current PhD candidate. Persister cells are a subpopulation of bacteria with phenotypic antibiotic resistance, capable of surviving antibiotic treatment and resuming growth upon antibiotic withdrawal. Persisters can cause chronic and recurrent infections in various organs, while also accelerating the emergence of clinically antibiotic-resistant strains. Studies have shown that macrophage phagocytosis significantly increases the formation of persister cells. However, the molecular mechanisms underlying persister formation within host cells remain poorly understood and controversial. Our team's research on Escherichia coli revealed that bacterial aggresomes play a regulatory role in persister formation. We subsequently demonstrated that bacterial protein aggresomes formation is driven by liquid-liquid phase separation. However, the role of bacterial aggresomes during viral infections remain unclear. This study demonstrates that RS stress in macrophages triggers rapid formation of intracellular Salmonella protein aggresomes, significantly increasing persister cell frequency. We first labeled the HSLU protein of Salmonella typhimurium SL1344 with EGFP as a biomarker for protein aggresomes. After infecting macrophages with these bacteria, distinct protein aggresomes were observed within the intracellular bacteria as early as 30 minutes post-infection. Further utilizing a mouse infection model, we also observed the presence of Salmonella protein aggresomes within macrophages in vivo. Mechanistically, macrophage-derived reactive oxygen species RS were identified as the critical inducer of protein aggregation. To quantitatively measure intracellular RS levels in bacteria at the single-cell level, we constructed a RS fluorescent sensor based on the RGFP2 protein. The results revealed that Salmonella-forming protein aggresomes were subjected to significantly stronger oxidative stress. To elucidate the biological functions of bacterial protein aggresomes, we employed single-cell fluorescence time-lapse imaging to dynamically track antibiotic killing and resuscitation of Salmonella, released from macrophages. The experimental data demonstrated that bacteria harboring protein aggresomes exhibit an 8.6-fold more likely to become persisters, establishing protein aggregation as a critical determinant in antibiotic persistence. We then performed high-resolution mass spectrometry analysis coupled with KGG pathway enrichment, revealing that these aggresomes are enriched with essential proteins involved in fundamental biological processes. We subsequently quantified ATP levels in individual intracellular Salmonella using Queen ATP biosensor, which demonstrated significantly reduced intracellular ATP concentrations in aggrosome-containing bacteria. To further elucidate the mechanism of persister resuscitation, dual RNA sequencing of Salmonella and macrophages was performed. Transcriptomic profiling revealed that during early infection, genes encoding the type 3 secretion system in Salmonella pathogenicity island 1 were significantly regulated. Intriguingly, bacteria with protein aggresomes exhibited higher SPI-1 gene promoter activity, suggesting that dormant bacteria retain infectivity. We further analyzed the dynamic changes in macrophage RS levels and their impact on bacterial recovery. The demonstrated a significant decrease in RS levels in late infection stages. This reduction triggered SPI-2 gene expression and aggresomes disaggregation, enabling bacterial regrowth. In summary, this study elucidates key molecular events underlying recurrent infections but also provides a theoretical foundation for developing novel strategies targeting persisters.